All right, let's dive right into what is, honestly, a fascinating molecular mystery. We're looking at four completely different drugs. Think of them as four totally unique keys. But as we're about to find out, they all somehow manage to unlock the very same incredibly complex biological lock, a pathway that can drive dangerous hyperinflammation. So our job today? Figure out exactly how they pull it off.
First things first, we need to understand the battleground. We're talking about hyperinflammation, what you've probably heard called a cytokine storm. This is basically your immune system going completely haywire. As you can see here, some kind of trigger makes immune cells release signals called cytokines. But here's where it gets really dangerous. Those cytokines then tell other immune cells to release even more cytokines. It becomes this catastrophic positive feedback loop, a vicious cycle that just spirals out of control. The real challenge is figuring out how to stop this storm without just shutting down our entire immune system.
So what's the central piece of machinery that's driving this whole storm? It's something called the IL-6/TH17 Axis. And the best way to think about it is like this. Interleukin-6, or IL-6 for short, is the master fire alarm. When it senses a threat, it blares this powerful system-wide signal. And that alarm call activates our heavy artillery, the TH17 cells. Now, these cells are absolutely critical for fighting off infections, but when that fire alarm gets stuck in the on position, they can cause some serious chronic inflammation. So this axis, this is our target. And this right here brings us to the core of the puzzle. How on earth do four drugs that seem to have nothing in common, an antimalarial, an antiparasitic, a really old gout medication, and a common heartburn reducer, how do they all manage to calm down this one specific pathway? It sounds almost impossible, but you know what? The molecular blueprints tell an absolutely incredible story.
Okay, let's put on our detective hats. It's time to crack this case by looking at the unique molecular blueprint of each drug one by one.
First up is hydroxychloroquine, or HCQ, and its strategy is so interesting because it's not a direct attack like you might think. It's much, much more subtle. Instead of trying to block a signal, it actually changes the physical environment inside the immune cell. It's literally altering the cell's habitat.
So here's the play-by-play. Because HCQ is what we call a weak base, it gets naturally drawn into the acidic parts of a cell, especially the lysosome, which is basically the cell's recycling and garbage disposal center. Once it's in there, it neutralizes that acid, which raises the pH. Now, that one little chemical change has a massive consequence. It completely jams up the cell's garbage disposal system, a process we call autophagy. And this is where you see the brilliant downstream effect. By jamming up all the cellular machinery, HCQ indirectly tamps down on those aggressive TH17 cells. But it also does something else that's pretty remarkable. It protects the peacekeeper cells, which are called Tregs. See, in some inflammatory states, that garbage disposal system can actually chew up and destroy the master protein that controls these peacekeepers, a protein called FOX3. So by jamming this system, HCQ protects this fire marshal, which helps bring the whole system back into balance.
Okay, let's move on to our next drug, Ivermectin, and you'll see a totally different picture. If HCQ's approach was subtle and indirect, Ivermectin's is a targeted strike. As you can see here, it's like taking a pair of wire cutters and snipping the connection on the genetic switchboard that tells the cell to produce that master fire alarm, IL-6, in the first place. So how does it cut that wire? Well, the switch that turns on the IL-6 gene is actually a whole complex of proteins. Ivermectin works by targeting one key piece of that machinery called PAC1, and it causes it to break down. Without PAC1 holding things together, the whole complex just falls apart. And that means the main command center, STAT3, can't even get to the DNA to flip the switch on. The impact here is huge, and it's way upstream. By stopping IL-6 production right at the source, you're essentially starving the whole system of its main inflammatory signal. Just look at the chart. You can see how ivermectin helps bring that critical balance back between the pro-inflammatory IL-6 and the anti-inflammatory IL-10. Less IL-6 signal means fewer of those TH17 heavy artillery cells can even be made. All right, on to our third drug, colchicine. And you guessed it, its strategy is different all over again. It doesn't go after the genes or the cell's environment. Instead, it targets the cell's internal panic button, this big protein complex called the NLRP3 inflammasome. Think of this as the cell's first responder system that triggers a massive,
rapid inflammatory response when it senses danger.
Colchicine's method is just beautifully direct. It's like throwing a wrench right into the gears of this machine. You see, the cell has all these inactive cytokine precursors just sitting around, waiting. The inflammasome is what activates them, turning them into their final, mature, inflammatory form. Colchicine just gets in there and physically blocks that from happening. Simple as that.
So how does this hit our central target? Well, colchicine doesn't stop IL-6 from creating those TH17 cells, but it takes away their performance enhancers. The signals it blocks, like IL-1 beta and IL-18, are what really supercharge the TH17 response. By taking those amplifiers out of the equation, colchicine dramatically weakens the destructive power of the inflammation that's already gotten started.
And finally, we've got our most surprising suspect, famotidine, a common heartburn reducer. The wild thing here is that its known mechanism, blocking histamine H2 receptors, is the whole story. The big surprise is simply that these H2 receptors aren't just in the stomach. Turns out they are all over our immune cells too.
So this introduces a really cool idea, a systemic primer. Histamine, when it hits these immune cells, gets them to produce a little bit of IL-6. It's like adding kindling to a fire. It doesn't start the blaze itself, but it gets the whole system primed and ready for much, much bigger inflammatory response when other signals show up. Famotidine works by blocking that receptor and basically kicking all the kindling away.
So the logic here is actually pretty similar to ivermectin's, just coming from a completely different angle. By cutting down one of the key upstream signals that drives IL-6 production, famotidine effectively reduces the amount of fuel, that master fire alarm signal, that's available to create new TH17 cells. It's another upstream strategy, just with a totally different mechanism. So now that we've cracked the case on all four of these drugs, we can take a step back and see the master plan. And what we find are two broad functional categories that emerge from these very different molecular blueprints. This diagram just pulls it all together perfectly. You've got ivermectin as a genetic transcription blocker, famotidine as a targeted receptor blocker, hydroxychloroquine as a cellular environment modifier, and colchicine as an innate immune emergency brake. Four completely unique strategies, all zeroing in to control that one central inflammatory axis. It's really incredible. And these four strategies really fall into two main camps. On one side, you have the upstream blockers, like ivermectin and famotidine. Their goal is to block the spark, to cut off IL-6 production and stop that inflammatory fire from really getting started. And on the other side, you've got the downstream modulators, like hydroxychloroquine and colchicine. Their goal is to control the fuel by managing the cell's response and limiting the damage after the fire has already been lit. And this all brings us to a final and, I think, really important strategic question. When you're facing a cytokine storm that's escalating fast, what's the better play? Is it to block the spark upstream to prevent that cascade from ever getting a foothold, or is the smarter approach to control the fuel downstream, working on the assumption that the fire is already burning and you just need to manage it? Connecting these deep molecular mechanisms to real-world questions like that, well, that's what makes this so important.